Hello, you guys. We're going to go ahead and move on in the Unit 1.3 notes. So in the last video, we talked about topic 1, which we kind of went over in general how enzymes work. In this video, we're going to look at topic 2, which is looking at different things that affect the activity of an enzyme. And so these are things that, different factors that are going to affect how an enzyme's able to do its job. Okay, and so there's a handful of things we're going to look at that are going to affect how enzymes are doing their job and whether or not they're doing it faster or slower, and catalyzing these chemical reactions. And so the first one I want to look at is the concentration of the substrate. So just to remind you, the substrate, that's the chemical that the enzyme is binding to at the active site. So in this diagram down here, these little blue diamond looking things, those are the substrate, and these red things are the enzyme, and it's going to bind to the active site on these enzymes. And when the enzyme is binding to the substrate, it's going to allow the chemical reaction to take place. And so if you have lots of enzymes binding to lots of the substrate, you're going to have a really fast chemical reaction taking place, because you're going to have all these enzymes binding to their substrate, chemical reaction, find another substrate, chemical reaction, find another substrate, chemical reaction. And so because of that, the concentration of the substrate will actually affect how fast the reaction's going. And so if you don't have a lot of substrate, like you have all these enzymes, but there's not a lot of substrates floating around, you have a low concentration of substrate, like here on the x-axis on the left side, that's going to be a reaction that's going to take place kind of slow overall, because you have all these enzymes, but it's gonna, they're floating around and these substrates are floating around, but since there's not a lot of substrates, they're not gonna be very often bumping into and binding to a substrate. It's gonna be harder for them to find the substrates to bind to them for the reaction to take place. But what's gonna happen as the substrate concentration increases, so now you have more and more and more, a higher and higher concentration of these blue diamonds, these substrates. Now these enzymes can way more easily find their substrate and bump into their substrate, because they're everywhere. So these enzymes are floating around, and then they bump into a substrate, they bind to it, chemical reaction, they float around, they bump into the substrate, chemical reaction. And so when there's a really high concentration of substrates, the enzyme's going to be constantly easily finding the substrate and binding to it, allowing the reaction to take place. Those reactions are going to happen very fast if there's a lot of substrate. But what's going to happen, though, at some point you're going to start seeing a leveling off. So if you look at this graph, the more substrate you have, the reaction starts to go a lot faster. The more substrates there are, these enzymes are going to have an easier time catalyzing this reaction if they're everywhere. But at some point, sorry, my mouse is being weird. At some point, though, you're going to see this leveling off, where even though you're adding more substrate, the reaction starts to not really go faster. And so there's a plateauing here where you're adding even more and more substrate, but the reaction's not continuing to happen faster. And that's because you've reached a point called enzyme saturation. And enzyme saturation is when you have a certain amount of enzymes that are present. And at this point, there's so much substrate that they're basically working at max speed. Like, they can't do their job physically any faster, because now they're not wasting any time finding substrate, because literally there's one everywhere. But now all the enzymes are working at max speed, taking a substrate, catalyzing the reaction, taking another substrate, catalyzing the reaction, taking another substrate, catalyzing the reaction. And that's basically going as fast as physically possible, because there's just so much substrate. And so now your enzymes at any given moment are all the enzymes, 100% of them, are catalyzing the reaction at any given moment, over and over and over again. And so the reaction has reached a maximum speed. Now, at this point, if you want the reaction to go faster, what you would do is you would just add more enzymes. So if you don't have enough workers working on this chemical reaction, these enzymes, the workers, just add more workers, and now that you're going to have more enzymes that are able to bind to more of the substrate. So that will help the reaction go faster. But when you have a set amount of enzymes, kind of like in this picture, you have 5 of them, let's say, theoretically you have 5 of them. At some point, once the substrate concentration is high enough, they're all going to be continuously binding and catalyzing the reaction, binding to the substrate and catalyzing the reaction, and they can't go any faster unless you add more enzymes. Then the reaction can continue going faster. So that's what's happening here in this diagram. So just know that the concentration of the substrate can affect how fast a reaction is taking place. Another thing that could happen is denaturation. So if you guys remember, or hopefully you know now, that enzymes are proteins that fold into a very specific shape. And when we talked about proteins, we talked about this thing called denaturation, where the environmental conditions can change in a way that causes the protein to start misfolding or unfolding or not folding properly. And that's going to be true for enzymes as well. Enzymes are proteins. And so if their environmental conditions like the pH or the temperature, if that's changed, they're going to be sensitive to that because they are proteins, and they're going to fold or unfold in a way that's going to make it hard for them to do their job. Specifically, it's the active site too that becomes really important. If this active site, this location where the substrate is supposed to bind to, if the shape of that is getting messed up because of the environmental conditions changing and the protein being denatured, well, then that's definitely going to affect its ability to catalyze the reaction. And you're going to see the activity of this enzyme and the speed of this reaction decreasing a lot when that's happening. And so so that's another thing that can happen to enzymes affecting their activity in cells. And so enzymes, depending on the enzyme, every enzyme has an optimal pH and an optimal temperature that it functions at, that it's going to do its job and catalyze the reaction most efficiently and fastest. And it just depends on the enzyme. And so when it comes to temperature, for example, for most chemical reactions, or just in general, chemical reactions in general, outside of biology, just chemical reactions, you should have learned this in chemistry, when there's a higher temperature, that actually causes chemical reactions to happen faster in general. If we're not talking about cells or biology, if we're just talking about chemical reactions, like one taking place on my desk when I mix chemicals together, that reaction is going to always happen faster when you increase the temperature. Because when you increase the temperature, molecules move around faster, and that's going to cause them to collide more often and the reaction to happen faster. Now in cells and in biology, that's also true. If the temperature is higher, it's going to increase the movement of molecules and enzymes and make it more likely that they run into each other. But you're going to reach a point where the temperature has increased too much that it's now denaturing the enzyme, and now all of a sudden that reaction is going to start very quickly slowing down. Because the enzyme is becoming denatured, and now the chemical reaction can't take place because the enzyme's not working. And so you can see on this graph two examples. There's an example of an enzyme found in a human being. And so for a typical enzyme found in humans, they actually function most optimally at like around 37 degrees Celsius, which is basically your body temperature. That's like 98 degrees Fahrenheit. And so most of your enzymes in your body, that's where they function most efficiently. That's where the protein, the enzyme, is folding correctly. Now, if it's too cold or too hot, if you move beyond that temperature, you're going to see a decline in the rate of the reaction. The reaction is going to go a lot slower when it's colder than that or warmer than that, and that's because of the enzyme structure, again, getting messed up. But again, that depends on the enzyme. Like I could take an enzyme from not a human being. Let's say I look at an enzyme from a bacteria that lives, a thermophilic bacteria. These are bacteria that live in very hot environments. And these enzymes, they optimally have a temperature that's way higher than the enzymes found in our bodies. And so let's say for these thermophilic bacteria, they have certain enzymes. Their enzymes usually have an optimal temperature around 75 degrees Celsius. That's way warmer than our bodies. And that's when they function most efficiently. And that's when they take on the correct shape. And if you're below that or above that, that's going to affect the shape of that enzyme, which could cause it to denature and cause that reaction to start going a lot slower if it's too hot or too cold beyond that. And so just know that every enzyme has an optimal temperature, and it depends on the enzyme, and it can denature when it's outside that optimal range. And same thing with pH. pH is another environmental condition that can cause proteins and enzymes to denature and misfold. And so every enzyme has a certain pH that it folds correctly. And so for enzymes, when they're folded correctly, they can catalyze that reaction the fastest when they're folded correctly and their active site is the correct shape. But when you change that and the enzyme starts to misfold or unfold and not be folded correctly, and the active site is not shaped correctly, you're going to see that reaction happening a lot slower. So here's a couple examples, both from humans actually. Here's the an enzyme called pepsin. This is an enzyme found in your stomach or produced in your stomach. And this enzyme has an optimal pH around 2, which is very acidic. But that's the environmental conditions of your stomach. It is very acidic in your stomach, and this enzyme folds correctly at that pH. And if you bring it to a pH that's more acidic or less acidic than that, that's going to cause that pepsin enzyme to denature, and that reaction is going to go a lot slower because of that. And then you guys also have another example of an enzyme you have in your digestive system is trypsin. Trypsin is an enzyme found in your intestines. And in your intestines, the pH is a lot different than in your stomach. This enzyme has an optimal pH around 8. It's slightly basic, actually. And so, and that's the environmental conditions that it's meant to fold correctly. And if you move beyond that pH, it's going to definitely affect the rate of the reaction, because it's going to affect the folding of that enzyme. So that's another thing that's, so those environmental conditions and the denaturing of enzymes can also affect how active they are. Another thing is mutations. And again, this is kind of something we've talked about before for proteins. You could have a mutation in your DNA that could affect the amino acid sequence in a protein. Let's say specifically this is an enzyme protein, and you have a mutation that causes that sequence of amino acids to now be changed. And so that sequence of amino acids is now different because you have a mutation. And so now that polypeptide, this enzyme, because of some of those amino acids being wrong, is going to fold maybe not the same. And if it folds differently, especially in a way that affects its active site and the ability of that active site to bind to the substrate, that's definitely going to affect the activity of this enzyme. And so there's some examples where you could have a mutation that is changing the structure of an enzyme because that mutation affected the sequence of amino acids that codes for this enzyme, and it makes it so that the shape of that active site is not compatible with that substrate, which is going to lead to that enzyme no longer working, for example. And so especially if you have, if it's a mutation that's affecting an amino acid in the active site of an enzyme, that's definitely going to have a high chance of negatively affecting that enzyme. If it's specifically in the active site region, one of those amino acids in that area, if one of those get messed up, that could definitely affect its ability to catalyze a reaction. Or it could be an amino acid not in the active site, but maybe somewhere else in the enzyme, an amino acid somewhere else away from the active site, but even that could still affect the folding of the protein and the shape of that active site. So that is another thing that could affect the activity of an enzyme. And then there's what's called cofactors and coenzymes. Cofactors and coenzymes, these are kind of like enzyme helpers. And these are molecules or substances that actually bind to certain enzymes and allow those enzymes to actually function properly. And so some enzymes, this isn't true for all enzymes, but some enzymes have to have these little buddies, these chemical buddies that they have to bind to. And when they bind to these chemical substances, they're now able to do their job. They're ready to do their job once they bind to these extra buddies that they have. And so those things are called cofactors, which bind, they're not proteins, but these are other molecules, substances that bind to the enzyme and then allow that enzyme to now start being able to catalyze certain reactions. Sometimes these are inorganic cofactors, which are like things like metal ions, for example, like zinc or iron or copper. You guys need small amounts of these in your body because those metal ions actually bind to certain enzymes that allow them to work properly, and without those metal ions, they don't work properly. And then there's organic ones, which are made of organic molecules, and we call those coenzymes. Coenzymes are organic cofactors. Really, for the purpose of this class, the difference between cofactors and coenzymes, you don't need to worry about it. Just know that cofactors or coenzymes, these are substances that bind to enzymes to help them work properly. And so an example of a coenzyme, which is an organic one, an organic cofactor, is vitamins. There's certain vitamins like vitamin A or vitamin D or vitamin E that bind to specific enzymes in your body that allow them to actually function. And without those vitamins, they're not going to function properly. So this picture kind of illustrates that. So here's like an enzyme that's been put together, but it's not until this cofactor binds to it that it's really now able to do its job and catalyze whatever reaction it's supposed to catalyze. Or here's another example, like this is the enzyme, this blob here. And it's only when you have this cofactor bound to it and this coenzyme bound to it, these other extra pieces bound to this enzyme, that it's now functional. And so for some enzymes, they're not fully functional until they bind to these cofactors or coenzymes. And if some of those are missing or not present, then that enzyme's not going to work. But again, that's not all enzymes. Some enzymes don't need any helpers. They just function normally by themselves. And then the last thing on here, which is very important, is the fact that there's inhibitors that can bind to enzymes and decrease their activity. And so there's these specific molecules that are called inhibitors, and they bind to specific enzymes. They can bind to specific enzymes. And when they bind to those enzymes, these molecules, when they bind to the enzyme, it decreases the activity of that enzyme. So that enzyme is now going to catalyze that reaction a lot slower, or maybe not at all, because that inhibitor is binded to it. So that inhibitor is it's going to bind to the enzyme, and it's going to affect its shape in a way where it's not working properly. And so there's two types of inhibitors. There's competitive inhibitors and non-competitive inhibitors. So I want to talk about each of these real quick. Competitive inhibitors, these are going to, again, reduce the productivity of enzymes by specifically attaching to the enzyme's active site and thus blocking the substrate from attaching. So a competitive inhibitor is like this picture right here. That's this blue blob, and it's actually binding to the active site of this enzyme. So here's the active site of the enzyme, and it's actually binding there, which is physically blocking the substrate from binding to this enzyme. So this is definitely going to inhibit and stop this enzyme from working because it's binding to the active site, preventing it from binding to the actual substrate. And so we call this a competitive inhibitor. And so a competitive inhibitor is usually going to have chemical properties that are very similar to the substrate that the enzyme's supposed to bind to. So there's something about this molecule that makes it compatible enough to bind to this active site. So this inhibitor molecule probably has some chemical properties that are in common with the substrate. They probably have some structural and chemical components that are in common because they both are able to bind to this area on the enzyme. But this is not the true substrate. This is an imposter getting in the way and blocking the real substrate from binding to the enzyme. So that's a competitive inhibitor. Now, a non-competitive inhibitor, this is an inhibitor, a molecule that binds to the enzyme, but not at the active site. So it's going to bind at a different location that's not the active site. But by binding to that different location, it causes the enzyme to change shape. So there's this enzyme, and when this inhibitor binds to it, the enzyme kind of changes shape a little bit when it binds. And that's going to mess up the active site shape, the shape of the active site, in a way that makes it harder or no longer able to bind to the substrate. And so that's what a non-competitor inhibitor is doing. And we call it non-competitive because it's not directly competing with the substrate for the same spot. He's binding to his own spot on this enzyme. Whereas a competitive inhibitor, he's competing with the substrate for the same location. They're trying to both bind at the same spot. Either way, though, these inhibitors are both having the same result. They are decreasing the activity of the enzyme. They're going to make the reaction go slower or make these enzymes stop being able to catalyze the reaction. Now, these inhibitors can sometimes be, they can either be reversible or irreversible. Most inhibitors are reversible, meaning that they just temporarily bind to the enzyme, and then they fall off sooner or later. They're not going to stay there forever. They have weak interactions with the enzyme. They latch on, but then they easily fall off. There's nothing permanently holding them onto the enzyme. So eventually, like, they're going to be there, but then they're going to fall off sooner or later. And then maybe another one will come and bind again, but like they're not going to be there forever. Whereas irreversible inhibitors, these are ones that are going to form actual covalent bonds, chemical bonds with the enzyme, and they're going to be stuck there permanently. And so in a permanent or an irreversible inhibitor, that's one that's going to latch onto the enzyme and permanently shut it down. And you could wait as long as you want, but it's not going to like that enzyme is now just done. Like it's not going to work ever again. Okay. And there's actually certain types of poisons. A lot of poisons actually function as irreversible inhibitors. And once they're intaken into your body, they're going to bind permanently to specific enzymes in your body that are really important and stop them from working. And that could cause death, like because they're no longer going to work. But like I said, most of the time inhibitors are just temporary. They're reversible. They'll bind, they'll go away, maybe they'll come back. And so the very last thing on here, and this is maybe the trickiest part about these inhibitors, is when we're looking at, let's just say we're looking at reversible inhibitors, so the inhibitors that bind to enzymes and then fall off eventually, and then they can bind again and then fall off or whatever. When we're looking at these reversible inhibitors, the competitive inhibitors that bind to the active site, we can kind of overcome them by increasing the substrate concentration. Meaning that if you have a really high concentration of substrate, if there's a lot of substrate, you're going to increase the chances that the substrate is going to bind to the active site before the inhibitor. So here's your enzyme floating around, there's some competitive inhibitors, there's some substrates, but if there's a lot of substrates, there's thousands and thousands of substrates, a really high concentration of substrates, and there's not a lot of inhibitors, then most of the time the substrate is going to bind to the enzyme first at the active site, and then the reaction is going to be catalyzed before the inhibitor gets there. Or if the inhibitor does get there, well, sooner or later it's going to fall off, and then again it's a race to the active site. And if there's more substrate, if there's a really high concentration of substrate, every new cycle there's going to be a greater chance that the substrate is going to bind to the enzyme instead of the inhibitor. And so really, theoretically, if you have a competitive inhibitor messing up an enzyme, if you just add more and more substrate, you can kind of overcome the effect that inhibitor is having on the enzyme, because you're going to give it a greater advantage to the substrate binding to that active site. Now, that's different if you have a non-competitive inhibitor. If you have a non-competitive inhibitor, it really doesn't matter how much substrate you have, because they're not competing for the same spot. So you could have lots and lots, thousands and thousands of substrate molecules that this enzyme is surrounded by. But if you only have a few non-competitive inhibitors, all they have to do is they just have to walk up. They just have to bind to this other location on the enzyme, and it's going to stop the enzyme from being able to work. And so it doesn't matter how many substrate molecules there are, because they're not competing for the same spot. Like once this guy latches on to his own unique spot, that enzyme's not going to really work. And so really adding more and more substrates not going to really help the situation, because they're not competing for the same spot. So in this situation, you can't really overcome this inhibitor by adding more substrate. And that's what this graph is trying to show you here. It's showing you 3 different lines where you're looking at, if you have more and more substrate, how the reaction rate is affected by an enzyme catalyzed reaction. Now, when there's no inhibitor, if you look at this purple line, this is kind of like the graph we saw earlier. If you add more and more substrate, the reaction will go faster and faster and faster. And then it starts to level off because the enzymes are becoming saturated, right? Now look at it if you have a competitive inhibitor. If you have a competitive inhibitor that's present, that's this red line, the reaction is going to go slower versus if there was no inhibitor. So if there was no inhibitor, the reaction goes really fast when there's more substrate. Now, when there's a competitive inhibitor, if you add more and more substrate, the reaction will continue to go faster and faster and faster, but not as fast as when there was no inhibitors. There is a difference here for any certain amount of substrate. If you had this amount of substrate right here, where my mouse is pointed, you can see, well, if there was no inhibitor, the reaction would be going this fast, which is a lot faster than if you had a competitive inhibitor, like right here, it's only going this fast. But you can see if you keep adding more substrate, you can overcome that inhibitor to the point where now it's going just as fast as if there was no inhibitor, because there's just so much substrate. But look at the difference here on this green line with a non-competitive inhibitor. When there's a non-competitive inhibitor, again, this is going to reduce the rate of the reaction because it's going to mess up the enzyme. But even when you add more and more and more substrate, you're not going to overcome this inhibitor. This inhibitor is going to bind to certain enzymes at a spot that's not the active site, and make it so that they can't catalyze certain reactions. So at any given moment, there's going to be some enzymes that have some of these non-competitive inhibitors bound to them, and it's not going to matter how much substrate you have, it's going to not be able to continue going faster. You can't overcome that inhibitor. So the reaction's still happening, just slowly, because there's these inhibitors getting in the way. And it just kind of sucks. You can't do anything about it because these non-competitive inhibitors, you can't outcompete them by adding more substrate. I hope that kind of makes sense, and the difference between competitive and non-competitive inhibitors, and how the concentration of the substrate can overcome competitive inhibitors, but not really overcome non-competitive inhibitors from affecting the enzyme and its activity. But that's basically it for topic 2 here. Only one more topic left in these notes, and that's the shortest one. So anyway, I'll see you guys in class or the next video. Thank you.